Hello everyone, welcome to Biology Board. This is Brijesh Chotalia. I am an MSc pursuer and the Department of Life Science. Today I am going to teach you about the Lac Operon. Lac Operon concept was discovered by Jacob and Monod in 1961. Let's go through all the slides of Lac Operon. So what is Lac Operon? Lac Operon is a set of genes which is responsible for the metabolism of lactose in E. coli. What is this? Set of genes responsible for the metabolism of lactose in E. coli. The Lac Operon was discovered by Jacob and Monod in 1961. The Lac Operon consists of three structural genes and three control genes. Three structural genes are responsible for the synthesis of three enzymes, namely gene Z, which synthesizes the beta galactosidase, gene Y, which synthesizes galactosidase permease, and gene A, synthesizes thiogalactosidase transacetylase. There are three control genes. Three control genes are responsible for controlling the Lac Operon system, which are gene I controls as a regulator gene, gene P acts as a promoter gene, and gene O acts as a operator gene. Let's say this is a DNA strand. Okay, this is a DNA strand. Here I have taken the notch because here it will be a operator gene, and operator gene has one notch where a repressor protein binds. Let's draw this as a whole. I, P, O, Z, Y, and A. These are control genes, and these are structural genes. Okay, this is a repressor protein. This is a repressor protein. It binds as a operator region. Repressor protein. When this repressor protein binds to the operator region, let's say this is RNA polymerase. RNA polymerase. If a repressor protein binds at the operator region, RNA polymerase cannot bind at the promoter region. And if RNA polymerase cannot bind to the promoter region, then the whole DNA cannot be transcribed. So, repressor protein acts as a repressor in operator region. The operator gene occupies a part of promoter gene. When the operator gene is active, the structural gene synthesizes enzymes. Here it is in inactive state because repressor protein is bind at operator region. But when it's active only when repressor protein is not bind at the operator region. So what happens? The activity of the operator gene is decided by a repressor protein which is synthesized by regulator gene. I this is I which is a regulator gene. It regulates as a on and off switch. Okay, repressor protein is synthesized by a regulator gene. When lactose is absent in culture medium of E. coli, the repressor bind to the operator gene and the operator gene is made non-functional. When lactose is absent in culture medium, let's say this is all a culture medium, okay? The repressor is bind at the operator region, okay? And the operator gene is made non-functional because the repressor protein binds at the operator gene, okay? This state is called as a repressed state and the phenomena is called as repression, okay? And what happens during repression? the enzymes are not synthesized because the operator region is occupied by a repressor protein. Okay, what happens during repression? Enzymes are not synthesized. When the repressor binds to the operator gene, the RNA polymerase cannot bind to the promoter. Oops, sorry. When the repressor binds to the operator gene, the RNA polymerase cannot bind to promoter and cannot unwind the DNA for transcription. Okay, when as we have seen, chal bhai chal. Okay, sorry. When the repressor is bound to the operator gene, this RNA polymerase cannot bind to the promoter region and hence it cannot unwind the DNA. And for the transcription, yes. Now, when lactose is introduced into the culture medium of E. coli, it diffuses into the cell and binds to the repressor protein to form an inactive repressor inducer repressor complex. Okay. Now, what happens if lactose is introduced into this culture medium? Lactose works as a inducer. Okay. This was the repressor protein. Lactose binds with the repressor protein. Okay. Lactose binded with repressor protein. Okay. Here. Okay. It diffuses into the cell and binds to the repressor protein to form an inactive inducer repressor complex. This was the repressor protein. Inducer, that means lactose. Lactose binded with this repressor and it obtains an inactive state. And this is the complex which is known as inactive inducer repressor complex. Okay, and this inactive inducer repressor complex cannot bind to the operator region as it gets inactive due to the introduction of lactose in it. It cannot remain binded to the operator region, and the operator region is set free to do its function. And this state of the operator gene is called as de-repressed state. Okay, what was first of all it was repressed. Now it is de-repressed because inducer binded with this one. Okay, and the phenomena is called as de-repression. Okay, let's get this here. Okay, here. The inactive inducer repressor complex cannot bind to the operator region and the operator gene is set free to do its function. The active state of the operator gene is called as de-repressed state and the phenomena is called as de-repression. Now, when the operator gene is de-repressed, the RNA polymerase can bind to the promoter gene and unwind the DNA and reads it. What happened? This is a DNA. Here it is a notch. Okay. Regulator, promoter, operator, structural gene Z, Y, and A. I, P, O, Z, Y, and A. This was the repressor protein which was binded here, but due to introduction of lactose in it, it came into inactive form and cannot get bind to the promoter region. Sorry, operator region. So now at promoter region, RNA polymerase gets bind. RNA polymerase, it functions as it unwinds the whole DNA. It unwinds the whole DNA and transcription process gets starts. Okay. So this initiates the transcription of structural gene. Why? Because the RNA polymerase binds to the promoter gene and unwinds the DNA and reads it. Now what happens during transcription? The transcription of structural gene leads to the synthesis of three enzymes, namely beta-galactosidase, galactoside permease, and thiogalactoside transacetylase. What are their functions? These three enzymes bring about the metabolism of lactose. As you can see, the definition of Lac Operon, what was the definition? Lac Operon is a set of genes responsible for the metabolism of lactose. Okay, so what will happen? Beta galactosidase, first structural gene, it will split lactose into glucose and galactose. Okay, galactoside permease, it facilitates entry of lactose into the cell across the phospholipid bilayer of the cell membrane. And the third one, thiogalactoside transacetylase. What happens in this? It transfers an acetyl group from acetyl-CoA to galactosides, lactosides, and glucosides. These are the functions of three structural genes. What does it do? How does it brings the metabolism of lactose? Okay, so in Lac Operon system, the lactose functions as an inducer for the synthesis of the three enzymes. As we have talked earlier, lactose works as a inducer. Lactose functions as an inducer for the synthesis of three enzymes. For the synthesis of these three enzymes, hence the Lac Operon system is also called an inducible system. The another name of Lac Operon system is inducible system. Okay. Now, the Lac Operon is a system of two types of regulation. We can see two types of regulation in a Lac Operon. First one is negative regulation and the second is positive regulation. How does negative regulation takes place? So, a repressor protein acts as a negative regulator. As we have seen, in negative regulation, the regulated protein repressor, seen as a repressor protein, we have it prevents gene transcription. As we have seen, when repressor protein was binded at the, let's do here again, when it was binded at the operator region, it was preventing gene transcription because it was not allowing RNA polymerase to bind at the promoter region. Okay, so it works as a negative regulation. Repressor protein, this protein works as a negative regulation. Then thereafter lactose was introduced into the medium. Lactose works as an inducer. It binded with repressor protein and the operator gene was set free to do its function. Therefore, RNA polymerase unwinded the DNA and transcription was carried out. Okay. Positive regulation. How does positive regulation takes place? The CAMP protein acts as a positive regulator. This protein, CAMP protein, this is the protein which acts as a positive regulator. What is so? What is CAMP complex? It is a cyclic adenosine monophosphate AMP. What is CAP? Catabolite activator protein. So this is the full name. Cyclic adenosine monophosphate catabolite activator protein complex. It functions as a regulatory element of Lac Operon. It works as a regulatory element. It also works as a switch on and switch off function. The CAMP cap complex binds to the base sequence in the DNA of the promoter gene in order to start transcription. So as we see, CAP complex, it directly binds to the promoter gene in order to start the transcription. So when the lactose is absent in culture medium and hence we have to do transcription, although we have to do transcription, then CAMP complex, it binds, is a protein, it binds to the promoter region and it starts transcription. So this is all about transcription. Thank you for watching it, and if you want this PPT, I will share you with it. I will give you the link in the description. Thanks for watching.